Welcome, everyone. So glad to have you here with us today. As you know, research in ALS is continuously progressing, and at Everything ALS, we are committed to uncovering new discoveries, supporting early detection, and working towards finding a cure for ALS. Would you like to help us by participating in our speech study? The purpose of the speech study is to improve the diagnosis and prognosis of ALS. We're looking for participants to provide 5 speech recordings, each lasting 15 minutes once a month, for a total of 75 minutes over 5 months. That's a short amount of time for a big contribution. We welcome both healthy participants and individuals diagnosed or suspected of having ALS to join us. To enroll, please visit everythingals.org/speech project, and that will be in the chat for you to copy. One other thing that I would love to ask you to help us with. We are actively recruiting for another study, the asymptomatic gene carrier study. If you're a gene carrier and asymptomatic and are interested in remotely monitoring your speech, cognition, and motor skills, please visit us at everythingals.org, asymptomatic study. That will also be in the chat for you. I have something very interesting to share with everyone today. It is a scholarship. What a wonderful family. The Segelman family is giving a $5,000, up to $5,000 for a scholarship. They are renewable for up to 4 years, provided that the student maintains a minimum of 2.5. An individual needs to be, who has faced financial hardships due to ALS diagnosis, either on their own or maybe a family member. You need to be at least 17 years old and live in the United States, and plan to enroll in a full-time undergraduate program. If you'd like to have more information about this, you will find the everylifefoundation.org/Segelman in our chat. And with that, once again, I welcome you all, and I will now turn it over to you, Christian.
Thanks so much, McFinn. Thanks, everybody, for joining us this evening, or evening on the East Coast, at least. And today's program, titled Fast Tracking Discovery in ALS, Collaboration, Innovation, and Open Science with Dr. Methylcobalamin, is about to start. But just before we get going, I just wanted to offer a little housekeeping. Just keep in mind that it's an interactive program. Merit is here to not only present, but also take your questions about the ongoing trials that are going and other innovations that are happening. You can ask your questions in the chat using the chat functionality. We will hold the Q&A at the end of the formal presentation, but there will be plenty of time to take your questions after that. With that, I'm going to introduce Merit. So Dr. Methylcobalamin is Executive Director at Mass General Brigham Neuroscience Institute and the Director of the Sean M. Healey and AMG Center for ALS, and the Julianne Dorn Professor of Neurology at Harvard Medical School. Dr. Cudkowicz is the inaugural Executive Director of the Mass General Brigham Neuroscience Institute, and her research and clinical activities are dedicated to the study and treatment of people with ALS. And Dr. Cudkowicz is one of the founders and past co-chairs of the Northeast ALS Consortium, NEALS, a group of over 150 clinical sites in the United States, Canada, and Europe, and the Middle East dedicated to performing collaborative clinical trials and research in ALS. She's brought innovations to accelerate the development of treatments for people with ALS, including having a senior role in the first antisense oligonucleotide treatment for a neurological disorder, which was SOD1, adaptive trial designs, and a central IRB, all with the goal to bring the best treatments as rapidly as possible to us. Dr. Cudkowicz is a principal investigator for the Clinical Coordination Center for the National Institutes of Neurological Disorders and Stroke, and the Neurology Network of Excellence in Clinical Trials, also called NeuroNext. Dr. Cudkowicz is launching the first platform trial initiative in ALS, the Healey ALS Platform Trial, a program that will greatly accelerate therapy development in ALS. She's received the American Academy of Neurology 2009 Sheila Essay ALS Award, the 2017 Forbes Norris Award for the International MND Alliance, the 2017 Pinnacle Award from the Boston Chamber of Commerce, and the 2019 Ray Adams American Neurological Association Award. Dr. Cudkowicz is a dedicated educator, mentors many young neurologists in clinical investigation of ALS and related neurodegenerative disorders. And she's completed her undergraduate degree in chemical engineering at MIT and obtained a medical degree in health science and technology program at the Harvard Medical School. She served her internship at Beth Israel Hospital in New York and her neurology residency in fellowship at MGH, where she's still working today. And she also obtained a master's degree in clinical epidemiology from the Harvard School of Public Health. Merit is a tremendous friend of the community and to our organization. She's been a great advisor over the years. So without further ado, welcome, Dr. Cudkowicz. Welcome back, I should say.
Thank you, Christian, for your kind words. And it's great to see everybody, some new people and some people I've met before. So let me just share my slides. And as you always have here, I won't really want this to be interactive. I'll maybe talk for about 30 minutes, but then I hope to have lots of time for questions and answers. I always get hard questions. I'm not sure I always know the answers, but I promise to look into them if I don't. So there's so much going on in the ALS world of therapy development. It's very hopeful and very exciting and moving at a very rapid pace. And a lot of it's being driven really by big data initiatives and a lot of the work by EverythingALS and other groups. I really firmly believe that we have to do everything we can to break down barriers or things that are slowing down progress, collaborate widely, and keep innovating. This is a hard illness to understand and find therapies for, but it's not an impossible one. And the more we work together and the more we share our data, the faster progress is going to be. So I want to tell you a little bit about some of those initiatives, as well as just an update kind of where we are on drug development for people with ALS. So first of all, one of the things that is really different in the world of ALS is how close foundations, people living with the illness, and researchers and the government all actually work together to try to find cures for this illness. And it's how we learn how to keep doing better, how we learn what to focus on. And that is so critical and so helpful for everything that we do. There have been therapeutic breakthroughs in ALS, many in the recent years, that give us hope that this is a treatable illness. And as I mentioned, the power of collaboration is moving this really fast. And we are really leaders in the whole field of brain therapeutics in innovating in trials of how do we get answers, cleaner answers, faster, and how do we promote real access to being these new therapies for our patients. So I always like to start with a story of a patient that inspired me, and that was, there's so many, but this was the very first person that I cared for, Susan, and she had the SOD1 gene that we now have a marketed therapy for. But I learned so much from her about how to be a physician, how to care for patients, and how to make sure that everybody living with ALS has an option to be part of research, to be part of trials, and how to bring a community together. So we really a global effort to try to develop this first gene therapy for this form of the illness. And what we learned from this, I'll show you a little later, but it showed us a couple of really important things. One is that ALS is treatable. There are people in those trials of this gene therapy that stopped progressing and got better. There's also a new biomarker that was discovered from this. That's something that changes in the blood that predicts who's going to respond to the drug. Those are all amazing breakthroughs in the field of ALS that help not just the familial form of the illness, but all forms of the illness. The other thing I really learned from her and so many people that I care for is that it takes a village to care for people with ALS and to learn from them as well. And all the centers really do have teams of physicians and healthcare providers that are working together to make sure that we meet all the needs of the people that we care for, but also that people can go to centers where they have options for research. And whether that's done remotely, through with foundations like EverythingALS, or done at clinical sites, they're really additive types of research, and it's really important that people have access to therapeutic trials, but also other types of research, whether that's speech studies or providing blood for DNA, so that we can really learn from everybody and share that data openly, so that we can really speed up drug development in people for ALS. And ALS is really a model for this. There are many other diseases in neurology that are looking for what we do, and seeing how it speeds up development, and wanting to do that as well. We're always adding programs to our clinic based on feedback from the people we care for. And we did, for example, hear from people that they want to be able to still have access to research, even if they live far from a center, or if they can't get into a center. And so we developed this house call program in New England, and are really training other places that want to develop something like that, so that people can have access to clinical research and trials and care in their home. We also developed, based on input again from people we care for, a program called the Parenting at a Challenging Time program, to help people with young kids approach how to share the news of ALS with their grandchildren or their children. And these are just two of many examples, but it's really that partnership with people and families with ALS and their physicians and the foundations that we keep trying to build programs to help people, while we're also simultaneously developing new treatments and ways to make sure that people have access to those new research studies. So there is a lot of progress, and I love updating this slide whenever I give a talk. So we do have 4 drugs that slow down the illness that are available somewhere in the world. Riluzole, Radicava, reshaping, which is another name from B12 or methylcobalamin. Those 3 drugs are for all forms of ALS, and then Qalsody, which is the gene therapy for SOD1. We need a lot more, but we do have options for people today that can help slow down the illness. And there's also a lot of clinical trials going on, and many, many more coming in fall, late and winter of 2025. I just list a few here. Cudkowicz has a trial of a gene therapy approach to increase this protein stathmin 2 that helps repair axon damage. And that trial is for all people with ALS. Spinogenix also has a study. It's a drug really led by a patient organization, Project ALS, that funded the science and then brought it to all the way to trial in people. And that is what we call a MAP kinase inhibitor, and that blocks certain pathways that cause motor neurons to die. So it's blocking that pathway. And that's in a phase 1 trial in people with ALS. RAPA, which is a company that has a neuroimmunology approach to try to block inflammation with a cell-based approach. Astrals, which is by Novartis, which is a drug that blocks inflammation in the brain, and many, many more. I'm happy to take any questions about active trials or things that are coming. We also have several therapies that have been in small phase 2 studies, and those are studies that are looking for initial evidence that the drug might have an effect, a positive effect on the disease course. And we have actually 4 drugs that have some positive effects in phase 2, and they're all going forward for phase 3 testing. And that's by companies Clean, Prolennia, and Neurosense. And then just this week, the paper on interleukin 2, or also known as IL-2, was published in Lancet Neurology, and then showed a positive effect on the disease course. So we do have some things in the pipeline that already have some positive data. As I mentioned, we have an amazing community, and many of you here on the call that are part of clinical research and part of the partnership to make sure these studies are designed well and answer key questions. And there's global networks. Kristen mentioned the NEALS consortium that we formed, but we work so closely with our European colleagues in NCALS and our Asian colleagues in PACT ALS so that we can learn from each other, not duplicate efforts, and combine our data so that we can, again, learn more about ALS. There's more than 300 companies in the ALS space. It's actually gotten very hard to keep track of all of them, but that's exciting and that's hopeful. And then there's many innovations about how do we test all these drugs so that we get the best ones to go forward. We do have some challenges, though, and this is where I think big data and AI technologies can really help us. And those challenges are, I break them up into two categories. One is that the diagnostic journey is still very long for people with this illness. It still takes about 12 months from first symptom to diagnosis, and people often see 3 or 4 physicians before they get to the diagnosis, and that is time where we're not providing options for treatments. And so there's a real push and a need to diagnose earlier. And some of the studies you've heard about before that are looking for pre-symptomatic carriers of genes are geared towards trying to develop biomarkers or tools to get earlier diagnosis. And then we really know that ALS is not one disease. And we know that from just following people and seeing their course. Everybody's course is different. And also from the genetics where we have many, many different genes that can cause this illness. And why this is a challenge is that when we, in our trials, take everybody with ALS, we might be perhaps missing treatment effects where a drug might work in a subset of people, not everybody. And we know from other fields, like in cancer, where if you can figure out the different biology and you can target drugs to the right person at the right time and the right biology, you're going to get a lot more successes. So sorting out this biologic heterogeneity is going to be really critical for us getting really impactful therapies. And we have a new initiative called the ALS MyMatch program that is trying to get at that heterogeneity and that better targeting of treatments to the right people in the right time, which I'll tell you about. So I mentioned the collaborative groups. There are these 3 trial networks that are working really closely across the globe. And each of them is working on collecting data in a different way, big data on people with ALS, and sharing that so that we can understand the disease better. And along the way, they've really been part of the development of these now 5 marketed drugs. I mentioned 4 of them before that slow down the disease course: riluzole, edaravone, the B12, and the tofersen or Qalsody. But there's also Nuedexta, which we know helps people with a symptom called pseudobulbar affect, but also with speech and swallowing. So one of the new initiatives that the government has funded is called All ALS. And we're working with EverythingALS in this project. It's led by Dr. Bowser at Barrows and Dr. Berry at Mass General and many, many other institutions. And it's trying to tackle those 2 challenges I mentioned. One is the late diagnosis, and the other is the heterogeneity between people, so that we can really improve the chance of success for therapies. So the first part is called the All ALS Prevent Study. And that is a study looking to recruit people who have a known gene that causes the illness, but they're asymptomatic. And that's the same study you heard about at the beginning, where we are working together on this to try to understand if there are blood or digital measures that can that happen before clinical symptoms, so that, again, we can try to diagnose early and intervene early. And the second part, called Assess All ALS, is finding people who have clinical symptoms of ALS and to try to understand, again, the biology, and to try to understand the heterogeneity and develop new biomarkers so we can have really smarter trials and more successive trials. So this is recruiting well. It's still open. Really, anyone can participate in this. There's centers all over the United States. And it was designed with the input from many, many people living with ALS as part of the NIH AMP ALS program. So it has that feedback, and it's made to be as accessible and easy as possible while being very informative. The other initiative that really came from a lot of people with the illness working closely with the ALS Association is a project called Think ALS. And this is targeted towards primary care doctors, orthopedic surgeons, ear, nose, and throat surgeons, the people where sometimes people with ALS go to first. And to try to decrease, again, the number of physicians people see before they get the diagnosis. And it's really more of an education, a training tool to have people think about ALS and refer to the neurologist quickly so that we can help tackle this one challenge. The other thing I just wanted to mention is that there are a lot of foundations in the ALS space. And the good news is that they are working together to try to fund some of those gaps of knowledge and challenge it. And so I work with a foundation, ALS Finding a Cure, that decided this year that they want to put their funds towards tackling these two challenges. I mentioned the early diagnosis and the heterogeneity, and it issued a call for grants to basically use AI and machine learning technology to try to answer some of these questions. So they're looking for proposals of how do you subgroup people by biology, how do you do drug discovery by repurposing drugs using AI, how do you develop biomarkers, how do you get earlier diagnosis. And so this is these grants are due next week. We think we're going to get a lot of grants from people who are really new to the field. We really tried to tackle the MITs and Caltechs and other engineering schools to try to draw in new people to think about the ALS. And they were really encouraging them to use open source data sets that are out there for many of the foundations, because there is a lot of data out there that many of you have contributed to, and we want them used to try to answer these big questions. So I just want to stop and go back a little bit to kind of what really opened up the whole field of ALS. And that was the discovery by Dr. Brown, my mentor, and a wonderful physician scientist, and many others, of the first genetic cause of ALS. This was in 1993, so it was a while ago, but it wasn't until this discovery that a lot of people started studying ALS. And so since that time, that first gene discovery, there have been now over 20 genetic causes of ALS identified, and these are in people with multiple family members. But we also, the field has really discovered also 40 other genes that might not run in a typical, family that you have one gene and one disease, but are complex genetics where having these genetic changes increases the risk of ALS. And every time someone discovers this, it gives new ideas into the biology, new targets, new models, and helps us understand the disease better. And so there are some themes coming out from the genetics that apply for all forms of the ALS and give us ideas of where to develop treatments. And they're shown here in the different colors. And some of the new drugs coming out this year are really tackling these three pathways. We've also learned from the genetics and from all these people studying ALS about the link between ALS and some other neurodegenerative diseases. And why this is important is because a lot of these diseases share the same biology of this particular protein, TDP-43, being located in the wrong part of the cell. It should be in the cell called the nucleus, but it leaks out of there into the surrounding fluid and then aggregates. And so it causes damage to the motor nerves for two reasons. One is that it's not doing its normal function in the nucleus, so it has loss of function. And then it's causing toxicity by aggregating in the other part of the cell. So there are a couple therapies coming out this year that are really targeting this pathway, that are disrupting the aggregates, are returning the TDP-43 into the nucleus, and will really test whether fixing this problem is going to have a big impact, not just on people with ALS, but also people with these other related disorders that also have the same biology. So many companies and academic groups working on this target. So I mentioned kind of the therapeutic breakthroughs, and I mentioned the SOD1, and I do think that this was a breakthrough for many reasons. And again, there was a big team really led by Tim Miller and other people at Biogen, Ionis, and many academic institutions. But it told us that this illness can be stopped. And while we all thought that was theoretically possible, it wasn't until this study that we actually saw it happen in people. I'm not going to go through all the data because I think Dr. Miller has been here and other people have talked about this, but I want to point out the two key things. One is that this gave us a new blood biomarker for ALS called neurofilament, or NFL. This is a protein that should be in your motor axons, but in people with ALS, when there's damage to the motor neurons, it leaks out and accumulates in the blood. And this drug, shown in green, lowered neurofilament level by 50% after 12 weeks of treatment with this gene therapy for people with SOD1, and it predicted who responded to the drug. And that's a breakthrough in any field when you have a measure like that, because it means you can do drug development in a much shorter time period. So that's huge. The other thing that we learned here is that sometimes it might take time for a drug to work. So this drug took about 8 months before we started to see some people really slow down in their progression, stop progressing. And then 10 months, we saw between 25 and 40% of people actually get function back. So it tells us if the closer we get to the cause of the illness, if we can shut off that biology, there is ability to repair. And that, again, has led to people working on repair pathways in ALS, seeing how can we accelerate that pathway. And there are, in fact, some drugs in trial coming up that are working on repair pathways. So the other exciting thing that has really come from this breakthrough of the SOD1 is the whole idea of manipulating or changing genes in the brain or spinal cord for people with ALS. The technology and the tools to do that are now here, and it's important not just for the people with the genetic forms of the illness, but really for all forms of ALS, because you can use that same technology to make more of a protein or make less of a protein. And so what we have now is actually many studies going on of different approaches to have an even bigger effect in people with SOD1. So we have one company, Unicure, that has a AAV approach to shut down SOD1. And this is a one-shot treatment rather than someone getting something every month like Qalsody. So that's enrolling participants mainly in Asia. And then there's another one by Regeneron that can be given every 3 months. So people are innovating already about how we can make it better for people. And then there's a prevention trial going on as well to try to stop ALS from starting in the first place, called ALS. So that's all in people with SOD1 ALS, but there's other genetic forms of the illness that are now having clinical trials. There's a very large trial of a gene therapy for people with the gene mutation FUS that's being led by Neil Schneider at Columbia. There's a couple companies, Trace Pharmaceuticals is one of them, and Cudkowicz is another one, that are developing gene therapies to target UNC13A. That's an important gene that increases the risk of getting ALS, and it also is very involved in that TDP-43 biology. So both those companies are going to be targeting all forms of ALS, not just the familial form. And then Dr. Schneider has developed an amazing program called Silence ALS. I think he's spoken here as well, where if someone might have one of the genes that's very rare, and maybe there isn't a company developing a drug for it, he is working with a nonprofit called NLORM to custom make that gene for people. And he's really worked out with the FDA a reasonably fast pathway to get that to people. And I've been working with him for one of my patients on that particular gene, and it is a very smooth, exciting process that he's now built, and that's funded by the NIH. So I also mentioned that these tools to turn on or turn off genes in the brain and spinal cord are being used for all forms of ALS. And this is just some of the examples out there. And I'll just say that there's many more. Of the 300 companies, there's many of them that have gene therapy approaches that are coming. So the two that are enrolling right now have the blue asterisks. So one is a company, Cudkowicz, QRL is their code. And this is a gene therapy that increases this protein stathmin-2 that I mentioned before, that helps your axons regrow. And so what we know is that when TDP-43, that protein, is not in the right place of the cell, you don't make stathmin-2 correctly. And stathmin-2 is really important for your motor nerve health. And so at least in the lab, when you help make it back again, make it normally again, those motor axons can repair. So that trial is enrolling in Canada and also in Europe, and they are taking people from the United States, and then there's many places in Europe as well. The other company that's enrolling is Amylyx. We know them from Relyvrio, but they have also a new drug. It's an antisense oligo or gene therapy that affects something called kelpain, which is important in some of the cell death pathways. And they are actively enrolling now in this new phase 1 study. Coming up is an interesting approach by a company, Vectory, and that's VTX, and they are using gene therapy approaches to deliver an antibody that targets the abnormal TDP-43. So they use the gene therapy to get into the motor neurons, and they deliver this antibody. They can get rid of the aggregate, and they are working towards getting to their first study in people with ALS. And the last one I'll just put here is by a company from China, Spinogenix, and their drug is called SNUG-01. And this is a gene therapy to increase a protein called TRIM2, which is really important for cell membrane repair. And they have some elegant studies that suggest that that protein is low in people with ALS, and then improving it could be, or increasing it can be helpful. And they've published on their first patient with that, and they are actively doing other studies. So that's just some examples of how what we learn in the people with the genetic form is helping all forms of the illness as well. So in my kind of last section, I want to go through a little bit about ways we're thinking about how do we go through such a big pipeline in a way that gives people access, gets clear answers, and helps us pick the best drugs to really focus on. And I've spoken here before about the Healey ALS platform trial, and I'll just update briefly on it. But I want to spend a little time talking about our new program called the Healey ALS My Match program, because I think it fills another gap that has become very apparent in the drug development pipeline. So the Healey platform trial and the MND Smart platform trial that's happening in the UK and Scotland are what I will call late stage trials. They're phase 2B or 3. So they're when we already know the dose of the drug, we know that it hits its target, and we're really looking at, does this work or not? And if it does work, can it go to approval, or do we need another study? And those are really important studies to do efficiently. But before you get there, you really need to do the work in what we call the phase 1, phase 2 space, where you're making sure your drug is safe and that you're getting to the brain, it's getting to the target, and that you know the right dose. And it's often hard for companies, small companies or academics, to get funded for that space. There's only a few places that fund that, like the ALS Association has a small grant for that, the Department of Defense does, but often venture capitals companies don't want to fund in that space. They want to wait until you have that data before they fund. And so we see a lot of companies spending years trying to raise money to get into that area. So we decided to build a program to try to solve that gap. So I'll start with the platform trial. And this was to address the gap that there were a lot of drugs ready for phase 2, 3, and that we thought that testing each drug one at a time was super inefficient. And we wanted to use this approach where we build the infrastructure to test many drugs at the same time. We share that infrastructure between the companies and the drugs. We share the data from placebo, and we can really cut the time in drug development in half. And we cut the cost also in half. And we can do this while also learning about the illness. We designed this with the input from our patient advisory group, our industry partners, our NEALS network, and with the FDA. And we launched this in 2020, and we haven't since then tested 7 drugs, and we have the answers to all 7 of these. So normally in that time frame, you might have gotten the answers to 2. So that's how you can speed it up, again, by sharing the data and sharing the infrastructure. And over 1,400 people have been part of the study and really contributed to this. We couldn't have gotten this trial off the ground without a major gift from Sean Healey, who we named this after, that allowed us to tell the first 4 companies, come work with us. We'll cover part of the cost of the drugs for the first 4. help us get it off the ground, prove that it works and it's efficient, and then the future companies would pay their costs, which would be much less than if they did a standalone trial. And so this actually happened as expected. For each of the drugs, there still is a small group that gets placebo. 25% with the rest getting active drug for the period of time that's placebo controlled, and then everybody gets access to the drug until we have the results. We picked drugs that affected different pathways for the first 5. The 2 of them working on different parts of the immune system. That was A and B. Clean, that works on energy metabolism and helps increase something called NAD that helps your cells make energy. Prolenia, that works on a receptor called the sigma-1 receptor that helps motor neurons live longer. And then the 5th one that worked on the cell death pathway. The 6th and 7th had the same target. They were different drugs, but they had the same target, which was this what's called the integrated stress response, which is part of that TDP-43 biology. And everyone who participated was very kind to also give their blood for whole genome sequencing, for neurofilament, for other biomarkers, and we developed these 2 apps during that as well, with the goal to try to see if we can move more trials to the home and easier for participants. So we have the results. So of the 7, 4 were clearly negative, A, B, E, and F. So obviously we would prefer G. We would prefer them to be positive, but we learned definitively that these drugs shouldn't go forward. 2 of them, regimen C and D, had some positive data, and they are both going forward to phase 3 testing. And I'll show you a little bit about that data. And then the last one, F, we had mixed results. We had some effect on strength and breathing, and it's still in discussion about what to do about that going forward. But for the regimen C, which is clean nanomedicine, while they didn't hit their primary outcome measure, which was the ALS-FRS shown here on the left, there was a benefit on longevity, and this was also seen in their other study in Australia. And when we really looked a long time, when we followed people out in the open label extension, we saw an even bigger effect. We also saw an effect on a small effect, but a small effect on neurofilament. And this is going forward and planning for a phase 3 trial. The predopidine, which was regimen D, had, when we looked at everybody, had a positive effect on bulbar function, so speech and swallowing, articulation. But in the entire group, it didn't meet on the primary outcome measure of ALS-FRS. However, when we looked at pre-specified subgroups, and this goes back to the concept I mentioned at the beginning, where not everybody is the same with this illness, biologically or clinically, we did see a positive effect in people who were earlier in their disease course and had a slightly more aggressive form of the illness. And so for this reason, they are planning their phase 3 trial, which should start hopefully soon, to enroll people who are earlier in the illness, so 18 months from symptom onset. And they're going to treat a little longer than we did in the platform trial. We had 24 weeks. This will be 48 weeks, and followed by an open label extension. And this will be a global study. So we did take a break after the first 7 and to look, like, is there anything we want to change in the platform trial based on what we've learned and what the field has learned? And so we did make a couple changes for the next regimens. And one is we decided to go longer. The initial study was 6 months. And we decided to go longer, not so much based on the Healey platform results, but more on the gene therapy results, where we saw that even in a drug that targeted the exact cause of the illness, it took the time to see the recovery. And so if one goes too short on the trial, and you can miss that, and we didn't want to do that, but we also didn't want to go too long. So this was a compromise at 9 months. And we're also seeing a few drugs now, the B12, the predopidine, riluzole, radicava, they all show that the effects are bigger the earlier you start the drug. So we've moved from 36 months to 24 months. So those are the big changes. And we've added a few biomarkers. And so going forward for regimen H and beyond, it'll be this new protocol. Because we're changing the design, we are going to what we call retire the data from the first 5 drugs. And why that's important is now it becomes part of that open database for people to mine. So all the data from people who are on placebo is now available through this open source database called PROACT. And also all the samples that people provided are available for people to study and learn more about the illness, and all the genomic data, the whole genome sequence is going to go into that big database called ALS Compute for access for researchers. So I mentioned that we wanted to dive into this earlier phase, because what we're finding is a lot of companies aren't ready to go into the Healey ALS platform trial or a late stage, because they don't know yet the right dose and if they're hitting their target. And those areas, there's no one really supporting that area. So we wanted to develop a program in that space, and it's what we call a phase 2A space. And this is where we see fields really make progress when you can identify the right people to get the right drug, and where you know before you go into the late stage trials that you're targeting the biology you want to target. And we also along the way want to really make it easy for people to be part of trial. So we're positioning ALS MindMatch in what I call the phase 1B or 2A space, and that results that are positive or where you know that you've hit your target, they can go forward to phase 3 standalone or into the platform trial or in the platform trial in the UK. But we've kind of in a way de-risked the drug and accelerated it going into late stage testing. So what will this look like? It's being led by Dr. Suma Babu at Mass General, and we're very fortunate to have gotten a grant from an anonymous foundation to get this launched for the first 2 drugs. So we're going to start at 4 or 5 centers. Everybody who's at that center will have the option to have every biomarker that we have around. They don't have to have it, but we're going to offer it. And that includes taking blood to make people's motor neurons in a dish, so we can screen drugs in your motor neurons, whole genome sequencing, neurofilament testing, other blood and urine biomarkers that exist. And then as we get drugs that come into the platform, we're going to try to match people based on their biology. So the simplest example of that is perhaps the genetic one. Like if someone carries the C9 gene and we have a drug that works on the C9 biology, only people with C9 will go in there. But you can also see this with markers, for example, of inflammation. If somebody has a lot of inflammation in their brain, which we can measure with a PET scan, and we have a drug that decreases that, we would use that PET scan or that imaging test to pick people for that drug. And so we're going to launch this. The first study should start in June, so very, very close. And we also made it, just like we did with the Healey platform trial, we told the first couple companies that work with us that we will cover part of their cost to get them to buy into this idea. Now, this is different than a platform trial. Not every trial is going to be the same. So, some trials could be, very short, and some could be a little longer. But they all do have to look at these really key factors, like are they hitting their biology? Are they getting to the right people? What's the right dose? Again, they can be short. They can be 3-month studies. They can be 6-month studies. They can be open label, meaning no placebo, or if there's a reason you need a placebo, they can be. But there's flexibility in design. So we're excited about this. They can also be gene therapies, repurposed drugs, cell-based therapies, novel small molecules. We think there's going to be great interest in this. We've spoken in a few meetings, and we think there's great interest from biotech and pharma for this approach. We've picked a couple people to be our biomarker experts. We are looking for other people to be our cell-based experts and our IPS motor neurons experts and our imaging experts, and we're forming a patient advisory group like we have for the Healey platform trial. So this is all in development, but we are ready to start with our first drug, hopefully in June. The applications from the companies are due this Friday. We've done informational webinars. We've issued the call. We've tried to make it easy. we'll have the review in June, and then the ones from this call will start hopefully in the fall. But we have pre-picked one to start sooner. So just end, and I hope I didn't speak too long, that I do think we've made a lot of progress in ALS. There's a lot more to do. I love being in this field for many reasons. I really want to make a difference for people living with ALS, but it's also such a powerful collaborative group, and you can see what happens when people row and sink and work together. It gives me a lot of hope for the next couple months and years that we're really going to have even more breakthroughs. I care for so many people with this illness and become so close. I just wanted to have a little shout out to Shalina and Ais and their family, and also Jeannie Goodman, her family, who have inspired me, along with many other people, and the group at Niels that I get to work with, and at Mass General, and all the foundations that help support our work and work together with us. Thank you. I didn't see any of the questions, so
That's okay. We've collected them as we went along. So thank you so much. And we've got a few different types of questions. So thank you for that wonderful presentation. It was really in depth, and you covered a lot of ground and kept it really accessible. So there are some questions about specific studies and just kind of your take on how they've resulted. So I'm going to start off with one of them. What are your thoughts about the potential benefits of Spinogenix SPG302, and what stage is the trial at, as far as you know?
Oh, sure. I like the biology, so it's what I put it in that repair category. In that it helps make new. They're called new dendrites, so new connections between neurons. They are early in development, so they have done a phase. I guess I would call it a phase 2A study in Australia. They have not yet reported out those results, but I believe it was 24 participants for up to 6 months, looking at biomarkers, looking at does the drug actually do what it says it's going to do, like increase of synapses. And you can measure that with electrophysiology and other things. So they, as you know, just announced a compassionate use study, getting that on the ground while they also work on the design of their phase 2B study.
Thank you.
All right, the next question is, what are your thoughts on IL-2 in the MIRACALS study?
Yeah, well, first of all, I'm super excited that the results finally got published. They shared them publicly maybe 2 years ago, but it took a long time to get out. I think it's positive. I mean, the theory behind IL-2 is that it increases these good T cells, the T regulatory cells that are anti-inflammatory, and so they showed that in a subset of participants, so people who did not have super high neurofilament levels, so people who are not super fast progressors, which was about 80% of the population, that they showed an effect on longevity that was quite large. Why it didn't work in people with a high neurofilament isn't that clear, but I do think they saw a fairly large subset that responded. And they showed that their drug increased Tregs, so they did what it's supposed to do. I don't know what the next step is for that drug, because it's a marketed drug for cancer. And sometimes you can change the label of a drug if you have a positive study. You can discuss that with the FDA about whether there's enough data to change the label so then it can become an official marketed drug for ALS, or whether the FDA or the other regulatory groups are going to want an additional study or not.
Did that start out as an investigator-initiated study?
Yes.
Was it repurposing?
Okay. Yeah. Great. There's no. I think the company gave the drug, but it was an investigator-initiated.
Got it. Thank you. Next question. Can you provide an update on what you and your fellow research colleagues in the U.S. are doing to mitigate research funding cuts by the current administration?
Yeah. So first of all, I think we have to advocate as loudly as we can. For ALS and for research funding in general, because ALS benefits from discoveries in other diseases as well. The Academy of Neurology, which is really the neurology advocacy group, is lobbying and meeting with senators and congressmen on both sides of the aisle to explain the impact of federal funding on all neurological illnesses. And so many of us are part of that initiative. Our hospitals and universities are similarly working together to advocate. and trying to get out messages of examples of NIH support that made a big impact. There's so many of them, but we all have to do it. And I was so happy when I saw that the Department of Defense money for ALS did not get cut at all, and while many other groups unfortunately did. And I don't know who lobbied. I suspect it's people in this group and other foundations that really lobbied for that, but it's so important.
Thank you. Yeah, couldn't agree more.
All right, the next question is, will the Healey Center engage in NU9 clinical trials, and are there any plans for it?
I would love to. So I do think, so I am an advisor for ACAVA. So just for disclosure, I have some stock options, I think, but I think they would be good for example, the ALS MyMatch. So they have to first do their phase 1 study where they test their drug in people without any illness for safety, single dose, multi-dose. But when they're ready for the next stage in people with ALS, I think it would be perfect for that kind of phase 2A space. Make sure the drug is getting to the target, the right population. So I'm excited about that program.
Thank you. Koya is working to launch a phase 2 trial this quarter, and that's a combination of compounds. Do you know if it includes IL-2, if there's anything else that we should know about the compound there?
Yeah, Koya includes IL-2 and another drug called abatacept. So it's tackling 2 parts of the immune system that seem to be revved up in people with ALS. And Dr. Appel has led a lot of that work and published on the first 4 patients where he saw good safety and good response. And now they're planning to launch, I would call it like just an early phase 2 trial. I don't have a start date on them. I think they haven't publicly shared any start date.
Got it. Thank you.
All right, next question is, what's the difference between all ALS Prevent and the ATLAS study?
Yeah, the ATLAS study is a therapeutic study in people who are gene carriers for SOD1. And so they will receive the Qalsody drug as a way to see if it can prevent the onset of the illness. ALS Prevent, all ALS Prevent is following people with any gene that might cause ALS. And there's no therapeutic intervention. It's really about following people with different outcome measures, whether they're digital or fluid, to try to understand what's the earliest change that happens in ALS to try to improve diagnosis, but also help design other prevention trials in those other genetic forms of ALS.
I'll take the next one. Are there any preliminary results from Prime C phase 3 yet?
Yes, no, the phase 3 trial for Prime C hasn't happened yet. So Prime C is by Neurosense, and it's a combination of 2 drugs, ciprofloxacin and Celebrex. They've done a phase 2 study and reported positive biomarker data, safety, and also outcome measures, and they are gearing up to do a phase 3 trial.
Thank you.
Next question is there a known pathway by which COVID or COVID vaccinations could cause ALS?
It's a good question, and I'd say that we don't know about either. There are some people studying this, and in particular, Dr. Avi Nath at the NIH is really he's a neurovirologist, and he's looking at this. So I'd say first, we don't know that they cause it, but I do think that there's reason to look at it. And some thoughts are, can viruses trigger like inflammation or an immune response that in susceptible people, maybe people who might have been at high risk for getting ALS or another disease, that tip them over or trigger it. And so I do encourage people who might think that their ALS was at least timed to either the infection or the vaccine to contact Dr. Nath. And you could do that through your neurologist, because he's collecting people's blood and studying this and really looking at it carefully.
Mary, in the chart you showed on some of the different proteins and genes being targeted, you showed TDP and 43 and SOD1 seem to be mutually exclusive, i.e. SOD1 ALS patients don't have the TDP 43 aberration. Is that how it's meant to be interpreted, or no?
Yeah, I think there's still a little debate on that, but several studies have suggested that those two forms of ALS, SOD1 and FUS genetics ALS, do not have TDP-43 biology. I've heard more recently that some people think that's not exactly true for SOD1, but so I think it's still a work in progress. But that goes back again to the point that ALS is not one disease, just like breast cancer is not one disease, that there's probably many different forms of it.
Next question is, do you have an opinion on the phase 2 trial of the ROCK inhibitor Bravo, and results of the phase should be coming?
I think it's a good pathway, but I haven't seen any results of it yet. But I think it's a good idea.
Next up is any results from the Fossadil study yet?
I don't. I'm not sure. That's a good question. Sometimes I'm just being careful of what I've seen publicly and what I've seen privately. I don't think there's anything published about that yet. No.
Got it.
What are your thoughts on the study at Sunnybrook involving the opening of the blood-brain barrier to deliver medications?
I think it's very exciting. So this is using something called focused ultrasound, where you can briefly open up the blood-brain barrier and let drugs in, which is a big problem for developing drugs for the brain. I think it's going to be very helpful for drugs that have to be given once or twice. I don't think it'll work for a drug you have to take every day, because you can't get the focused ultrasound every day. But let's say there's a gene therapy. Right now, we might have to inject it in the spinal fluid or in the ventricles in the brain. It's complicated. But if you could do it in the blood and open up the blood-brain barrier with the focused ultrasound for a couple minutes, get it in there, and then it shuts back down, that could be a game changer for gene therapy delivery for all forms of ALS.
Does sound promising for them. As we study people with ALS, is research finding that most ALS has a genetic component?
I think it is. I mean, I'm not a neurogeneticist, but when I listen to Dr. Brown and Amara Al-Shalabi, they talk about these twin studies where they have followed people, identical twins, pairs of identical twins. I forgot if it's 25 or 30. And from seeing whether they both get ALS or just one and not the other, or 50-50, they've really come to the conclusion that about half of the cause of ALS is probably genetic. It's not the simple, one gene you got from a parent that causes the illness. It's probably complex genetics where there's multiple different gene changes that increase your risk. So that's the current number that I've heard, that they think about half of the reason somebody has ALS is in your genetics, and the rest is kind of environmental.
Is there any chance that the 2-year mark to reject patients into trials will be removed at some point soon?
I don't think so, and I'll tell you why. I think we have to find other ways so that everybody has options. I think we're seeing more and more trials go earlier. For example, in the platform trial, we went earlier from 36 to 24, and the reason is twofold. One is because some studies are showing us better effects early, and also that if you take people who are up to 33 years, you are enriching for people who are slow progressors, which is great. We want everybody to be a slow progressor, but in a trial of 6 months or 9 months, it means someone might not change in that time period, and it makes it much harder to see if your drug works. So the goal of the trial is really, with the fewest number of people, find out if it works or not, so you can get it on the market for everybody. And as you saw from the Polenia trial, they're actually even moving to 18 months from symptom onset. So I think we have to have other options for people who are past the 2-year or the 18 months mark, and that would be that compassionate use option, so that people can still have access to the drug and the therapy and be monitored safely, but that you're using the data from the people earlier in the illness to figure out if the drug works or not. So then you can do a shorter trial to get that answer.
Thank you. Can patients with bulbar ALS participate in the research? And if yes, is there a link to share to register again?
Yeah, yes, absolutely. I mean, almost all studies take people with bulbar onset ALS, whether that's the All ALS, natural history database, or the current clinical trials. So I don't have a particular link, other than I'd say that there's, defined clinical trials. There's there's a government website, clinicaltrials.gov, but there's also, I think, ALS TDI has a very good website for clinical trial finding, and so does IMALS. And obviously, your neurologist should be able to tell you as
well. I'm going to jump in on the next one as well. Why are the Canadian neurologists not giving the vitamin B12 methylcobalamin in the same way that it's being given elsewhere?
Well, I probably can't speak for the Canadians, but I'll tell you that this is the challenge. It's approved in Japan. Like there were 2 studies. The first study was with the broad population, and they actually did not see benefit, but they saw in a post hoc analysis where they looked at, it was their subgroup that responded. They saw a very big response if you started it early in the illness, between 12 and 15 months of your first symptom. Then they did another study just in people in the first 12 months, and they saw that same effect. So based on those 2 studies, it got approved in Japan. It's not approved in Canada or in the US, and that's not because Health Canada or FDA said no. It's because the company, Eisai, has not filed for approval. I think if they filed, it'd probably be approved, and then it would be covered by insurance, and everybody would be prescribing it. So my guess is they're not prescribing it because it's not a Canadian Health Authority approved drug.
Thank you.
What focus areas and organizations are doing the most exciting work leveraging AI for ALS research? Are there, specific sub-uses or areas that you think are particularly interesting?
I think it's going to be really helpful for this subgrouping of people. I think we now have a lot of data out there through Answer ALS and other groups on the biology from samples from people with ALS that we could start to subgroup people. There are some phenomenal scientists at UCSF and at MIT and in our group and Hopkins who are real experts in AI who are starting to mine this data. I know Vision 2030 is very focused on AI and supporting that type of work with EverythingALS. Now ALS Finding a Cure is trying to jumpstart, by funding some people and attracting new people. And I believe there's going to be a grand challenge. I forgot the name of the group in Europe. There's an organization that's going to give a big prize to someone who uses AI to solve some of these big challenges. So it's coming, but there's many groups working on it.
You mentioned PET scans to identify neuroinflammatory process. Can this be indicated for a newly diagnosed ALS patient to indicate possible response to steroids?
Well, we can definitely do it in people who are newly diagnosed, and a lot of centers have this. It's a PET ligand called TSPO. You could look at response to steroids with it. There have been studies of steroids in people with ALS, and they have not worked. So I'm not sure that would be the best anti-inflammatory approach, but you can use other anti-inflammatory drugs and that PET scan to see if you decrease that neuroinflammation.
Thank you.
What are your thoughts on testosterone and the role that it plays in ALS onset in men? Do you think that there's a connection? And if so, would that impact treatment options?
It's a good question. I honestly don't think we know enough about it. At least clinically, what I tell people I follow is that if their testosterone is low, we should definitely replace it. I don't think we should give it if someone's testosterone is normal, but if it's low, we should give it a try. But I think we need more research on that in general.
Are there any results from the, I'll see if I get this right, Ibolalast study?
Yeah, not yet. No, I think it was supposed to finish enrolling in April. I don't know if they've actually finished enrolling, and then it'll be about a year from when the last person enrolls. So I don't think we're going to know for a while. The previous study, we did a small study, but it was open label, and it was looking actually at the PET scan. We were looking at that measures of inflammation, and we did not see any change in inflammation or change in neurofilament. But it was small. It was 35 participants over, I think, a 6-month period. This study is much bigger, and they're looking at different outcome measures, but I don't think we'll know till 2026. unfortunately.
Thank you.
What's your opinion on how soon drugs will have FDA approval based on all the information discussed in the current pipeline?
I hope we'll have some in 2026. Nobody really knows what's going on in the FDA. So I hope that we're going to have that flexibility that we had with the prior administration. Right now, they're very short-staffed, and things are taking longer to get back from them. But hopefully, that will settle out. So for that reason, I think it's less likely to have something new this year. I think if Eisai would file that might that B12 that might get approved quickly. But I don't know that for a fact, but that would be my prediction.
We have gotten through most of the questions at this point. So this last one here is: do you have ALS patients with neurofilament levels that remain normal throughout their disease?
Yes.
And if so, yeah, what are your thoughts on why? Why might
Yeah, I do, and I saw Lyle's on. I'm curious if he does too. But yes, it tends to be in people who have a very slow or very isolated form. Like there are some people where it's only in the arm area or only in the leg area, and the progression's very slow. It always makes me a little nervous. Am I missing something? So I do tend to really look for other causes. But it has been reported, and it might be that the damage is just so minor that it doesn't leak into the blood. But we definitely see it. It's a good. I take it as a good sign when people have that.
Yeah. Dr. Cudkowicz, as always, thank you so much for not only presenting the updates you presented, but also joining us for the conversations and for making yourself so accessible to the community.
Thank you. Thanks for having me. It's really good to see everybody. Yeah. Have a good rest of your evening.
Yeah. Thanks. McFinn, we're gonna give it back to you now.
Well, thank you everybody for your attendance. Thank you, Merritt, for making us all feel like somebody is in the office late at night, making sure that everybody else is doing everything that they can to make sure that ALS is a memory. Folks, we need that. And we all know it. So let's all come together and help Merritt, help all of the doctors who are doing this research, make ourselves available for trials, and showing up for these type of talks is just showing that we have power in numbers, and that a suggestion from maybe one of us is going to become the answer. The doctors have been working on this for over 100 years. And so I think we need to get together and be detectives and do as much work and give the doctors as much information as we can give them. I know sometimes it's difficulty, especially if you have to travel to go to your clinic. But you know what? Tonight you didn't have to travel to go to your clinic. And Dr. Merritt came right to your home as a what do you call that, a house call? Those are the kind of things, folks, that we try to give to you because you're all very important. And it's only because of you that we're here.